And in this video we're going to look at how proteins add functionality to the membrane and allow for the passage of molecules, ions and information. There is a problem. How do we get material such as nutrients into the cell? How do we get the various molecules and ions and also information into the cell so that it can survive? And how do we get material such as metabolic waste out of the cell? Well, all of this is done by the proteins that span the membrane. In terms of transport across the membrane, it can be split into two types: passive transport and active transport. In passive transport, the movement of molecules or ions goes from a high to a low concentration and doesn't require energy. In active transport, the movement of molecules or ions goes from a low to a high concentration and requires energy to drive the movement. The proteins that are involved in the transport process can be viewed as carrier proteins or channel proteins. The carrier proteins interact directly with the material they are transporting. In contrast, the channel proteins form a pore through which the molecule or ions flow, and this process is regulated. The regulation may be in the form of voltage-gated, that is, the pore only opens when there is a specific charge across the membrane. For example, these are used in nerve and muscle cells. Regulation may be ligand-gated. That is, the binding of a ligand, a molecule, to the pore causes it to open, and this binding can occur inside or outside the cell. And the pore can be mechanically gated. That is, the pore is selective as to what it allows through. My favourite example of a carrier protein are the glucose transporters, which are called the GLUTs. Imagine my hands are a glucose transporter. The GLUT sits in the plasma membrane and flips between two states. In one state they are open to the outside of the cell, and in the other state they are open to the inside of the cell. At no point is there an open pore between the two sides of the cell. In the presence of glucose, the flipping occurs more rapidly. And so what happens is, in the open state to the outside of the cell, the glucose binds, and then the protein flips to the inside of the cell. And now the glucose is facing inside the cell and can diffuse away from the GLUT. And that is how glucose gets inside the cell. Now, one thing to note about the passage of ions across the membrane is the process is not only regulated by the concentration gradient of a particular ion, but also by the charge state of the cell. So, for example, you could have a very high concentration of a negative ion outside the cell compared to the inside, but if the inside of the membrane is carrying a high negative charge as a result of the concentration of other negative ions inside the cell, then you will not get movement of the other negative ions across that membrane. For active transport across the membrane, the energy required can be provided in one of three ways. The first two ways are fairly obvious. The cell could use ATP or light to drive the trafficking, the movement of the material, by causing a conformational change in the transport protein. The third way is not so obvious, and that is coupled transport. In coupled transport, the cell uses the transport of one molecule from a high to a low concentration to drive a conformational change in a protein. That conformational change then moves a molecule against a concentration gradient. As I said, this coupled transport approach is not so obvious and does need some thinking about. An example of this type of transport is the sodium glucose carrier, which is different from the GLUTs I mentioned earlier, as the sodium glucose carrier is moving glucose against the glucose concentration gradient. And this is an example of a simple coupled carrier. With the sodium glucose carrier, you have a high concentration of sodium and a low concentration of glucose outside the cell. And you want to transport the glucose into the cell against its concentration gradient. The transport protein uses the sodium gradient to co-transport, symport the sodium and the glucose into the cell.